All right, so let's do our study guide then. 17.
Can you explain the olfactory bulb neurons? I don't understand that. Like, is that like a structure or is it something inside it?
It's a neuron. Let me pull up that picture of everything real fast. So that's 17. It just says to label it, right?
Well, no, it's like the definition on.
Oh.
Describe the function. Yeah, describe the function.
Yeah, so its function is to take the signal. Wait, where's my stick? The function of the, 17B. 17 part 1B. So the function of olfactory bulb neuron. And its job is to take the signal from the olfactory neuron, like the receptor cell, to the brain. Because you've got your receptor cell in, what color is that? Forgot already. In blue here, but it only takes the signal to the olfactory bulb. But then the signal has to get to the cerebral cortex, so you know what it is. So the bulb neuron takes the signal from the bulb to the cerebral cortex. Want to make me do that one again? The steps of olfactory transduction. So there they are. And this is the thing that you did the model for. So just do the model again. Get some Play-Doh. So honestly, you could write these words over and over again. And unless you do something with them, does it make any sense? Do you think it makes sense if I just say them again and again? It's not. So grab some Play-Doh, some popcorn. Just basically do what we did in class a few times, and you'll have it. Yeah, for now, I guess.
And what about explaining how different olfactory receptor cells are activated by different organs?
So it's all about the shapes being different. So different odorants. This is G on part 1. So different odorants have different shapes. So the receptors have different shapes too. And they have to fit into each other to get activated. Yeah, so we've got, do you remember what activated? Oh, sorry. So this is I on part 1. How did the salty taste buds get activated? So sodium ions went into the salt taste buds. H plus ions just went through the sour taste buds, through channels, kind of like we did. And then the other ones, so like sweet, bitter, umami, those are kind of like the odor, where there's like a receptor and the tastant hits the receptor and activates it. And one thing to kind of remember is for all the taste buds and all the olfaction, all the steps, eventually you get to depolarization, where like positive ions come in, and that's what activates the action potential, and that's what sends it all along the axon. That's kind of a similarity for all those, because they are neurons. So. Anything else on part 1?
So was for the, like, B and E similar, like describe the function of bonds, the supporting cells, and the receptor cells, and beta cells, and it was basically the same thing, just different.
Right. So like you'd say the olfactory receptor cells, sense the odorants, and then the gustatory receptor cells sense the tastants. So yeah, they're very similar in function. It's like the basal cells are the same, yeah.
The support cells are just, like, for investigation, it just said that they surround the gustatory receptor cells, so these are literally just there as like.
They're there as support. So the support cells, yeah, just in their name.
I used to just don't understand like what kind of support.
Mostly structural support. Maybe a little bit. But really the supporting cells for olfaction and gustation, they're just there for structural support. That's really all I need to know about them. You want to do part 2? Yes. Part 2.
I just don't, is that just what you want to, like, how it goes in with the light? Or is it more like the structures, like how it affects?
Yeah, so C would be, so part 2, C, yeah, the structures it goes through.
Okay, so yeah.
So if we pull up a little picture here. So it would go through the sclera, the cornea, the pupil, the lens, to the retina. So we don't actually, not the sclera, sorry. So cornea, and then it goes through the pupil, the lens, to the retina. So cornea, pupil, lens, retina. Well, for everybody but you. Yes, so for D, explain how photoreceptors transduce visual information. That's the in the light, in the darkness. Although I guess really it's just in the light. Where is it? So that'd be like that diagram that we practiced on a whole bunch of times.
So just explain those steps.
Right. You could look at that, or you could look at the tables you filled in for like when it's actually activated. And of course, what was the confusing part about this? Is that when you're in the light, that first cell is like turned off basically, which means the second cell gets to be turned on because it's not being told to be off. It's like backwards. That's that one. You're right. So H, explain the path sound waves take to get to the cochlea. So, here's your ear. It goes through the auditory canal to the tympanic membrane, which is the eardrum. Then it goes through a couple bones, and then it goes into the cochlear fluid in there. You don't need to know which they are. Just know that it goes through these 3 bones here.
What was the first one?
It goes through the auditory canal. So auditory canal, tympanic membrane, a series of bones, and then into the fluid of the cochlea. Oh, for E? Yeah, so contrast the different types of cones. Well, the difference is which kind of light activates them. Or I guess really turns them off, right? So red light turns off red cones.
What color?
Mm-hmm. The photoreceptor cell. Mm-hmm. That's this one, the one we just did. The one we just did, D, how photoreceptors transduce visual information. It's okay. So it's that table you filled in, and then or that describing that diagram. It's the same thing. What is different about different colors of light? Wavelength and amount of energy. Anything else for 17? Did anybody write down how many points I told you it was going to be from 17? I remember 20, 80. Okay. So 20 points from 17, 80 points from the rest of it. Let's see, 1, 2, 3, 4, 5, 6. I think 2080 is a good mix. I may have said something different, but.
No, you said both, but
Oh, well, I couldn't either, so. And one thing, though, for anything that has a part 1 and part 2, it's like directly proportional. So if we spent 1 day on a chapter, that chapter is going to have less questions than the chapters we spent 2 days on. So it's kind of proportional, the amount of stuff that's on here. All right, so what about chapter 1 then? The major functions of all 11 systems. I I guess in the interest of time, I'll just send you back to the book for that one, or the PowerPoint. So that'll probably take us like 5 or 10 minutes to go through all of them. But make sure you're keeping it simple. So you don't need to tell me, all 8 functions of the skeletal system. Like, for example, what's the main function of the skeletal system? Support, help with movement. So keep them simple. If you look back at your study guides and you've got like a paragraph for each one, don't try to study all that. Just simple for this. Anything else for part 1? Or chapter 1?
Oh, can you do the components of the feedback system?
Yeah, so the components, you've got the receptor. So sorry, this is A for chapter 1. The receptor senses the change. It tells the control center about it. So you got so the receptor detects the change in the condition. The control center decides what to do about it, and it sends a signal to the effector, which fixes the problem.
Can you repeat them, please?
So negative and positive feedback.
So, the negative is just a change in temp, right, in your body, is that right?
For an example, that would be one.
And then positive would that be positive?
Yeah, so the example of positive feedback is like the uterine contractions. They just get stronger and stronger until the baby's out. And then the difference between like that idea is so if it was negative feedback, the response of the system is going to turn off the stimulus. That would be negative. That's how most of them are, like body temperature. Your body temperature is too low, go through these steps, you increase the body temperature, now the receptor's not turned on anymore because the temperature's back up. But if you're doing positive feedback, this response, this is positive, it's going to activate the receptor more. So for example, the childbirth, the baby pushing on the cervix tells the receptor to tell the uterus to push harder, which pushes more on the cervix, which tells the receptor to tell the control center to push harder and harder. And so as you're pushing harder, you're telling the receptor, like, you're getting it stimulated more. So positive feedback is the one where you're just like repeating yourself over and over again. Whereas negative feedback, it's like you're turning off the stimulus, you're fixing the problem. And negative's a lot more common. Most of the systems are negative feedback. You wouldn't want your body temperature one to be positive feedback, because then you'd just keep getting hotter and hotter or something. It'd be a real problem. All right, what about chapter 3? All right, chapter 3b, which method of transport? Do it. If you've got the flow chart, that's perfect. All right, so for this one, it'd be like I'd give you a situation, like this molecule going from this concentration to this concentration. whatever. And if you remember, we had like a flow chart that we came up with to decide which kind of transport it was. Does anybody remember the first question? Or I'm going to sneeze in a second. Sorry. All right. So is water moving? If the answer is yes, what are we talking about? If the question doesn't ask you about water moving or something, we got no. And we got another question. Well, you could ask it either way. And if the answer is yes, active. And then we need one more question. So is it charged or large?
Yes,
We're facilitated. All right, so for example, you've got this flow chart, and the question was something about sodium ions going from 10% to 2%. You come over to your flow chart, and you'd say, is water moving? No, we're talking about sodium moving, so we're here. Is it moving from low to high concentration? It's not. Is it charged or large? It is, so you're at facilitated. So really what you'd want to memorize is the little flow chart thing. Does it matter if it's moving inside or outside the cell? Do you see any questions like that up here? No. So all these, active, simple, facilitated, it could be going inside the cell, it could be going outside the cell. What matters is what's moving and if it's moving from low to high or high to low. Do you see anything that says hypertonic or hypotonic up there? That's not a kind of transport. Don't write that. People always do. Don't know why. So I'll say what kind of transport, and they'll say hypotonic. So don't do that in your exam, please. Yeah, so the direction of osmosis, chapter 3a. So which way does water go? So it goes, well, we're just looking for which way water's going to go. So does it go to areas of high solute or low solute? So it goes to the area that has higher solute concentration. That's all you have to know for that one. So if, there's some picture that says 10% calcium, 5% calcium, which way is water going to go? So it's going to where there's more solutes, water in. So it'd be a question like that. Now, if it's written out like a description of something, do you think maybe you should draw yourself a little picture on your test? Because I guarantee you, if you actually draw that, you won't miss it. If you try to see it in your mind and just put your answer, a lot easier to just get the numbers backwards or something, which would be sad. Sorry.
C, but so the DNA to RNA
Yeah, so central dogma from molecular biology, DNA to mRNA is transcription, mRNA to protein is translation. Simple as that.
Which one's translation again?
mRNA to protein is translation. DNA to mRNA is transcription. Right. Chapter 4, part 1.
Or, I mean, I get what they are. I just was, like, there was not really a definition of what I just didn't get the nervousness of what it actually.
So yeah, for D, list and briefly describe the 4 types of tissues. Which ones did you want? Muscle and nervous?
Yeah.
So muscles for movement. Nervous is to transmit signals.
And then the other 2 are epithelial.
Epithelial is like barriers. Connective is to connect things together. What else? See, describe the 3 functions of epithelial tissue. So 3 main functions. It's a selective barrier. It protects from abrasion, like, physical things rubbing against it. And then secretion. All right. What about part 2? Or part 2?
For B, are you gonna want to know more of the, like, where it would be or what it does? Because I mean, I know it asks for both, but
Yeah.
I'm probably gonna ask both.
I don't know which one I'm going to ask about. Now,
You just know all of it.
so this is describe the main function and give an example of where you'd find each of the following. And it's in part 1 and part 2. You don't have to list all the locations. Just be able to tell me one. And then the main function. Like, what were most of the epithelial tissues for, or a lot of them? So a lot of the different epithelial tissues were secretion and absorption, right? And then, or their protection. And you probably have all those answered on your study guide from last time, I hope.
Yeah.
But yeah, pick one location, describe its main function, keep it simple. What about 5? The skin chapter. Do I? So, A, explain how the structure of the epidermis supports its function. So you could take a few different routes on that one. I guess we could do 2. So one of the main functions is, protection from abrasion, basically. So when you rub on something, you don't rub off your muscle and your blood vessels or something. You just lose some skin cells. And its structure supports that because it's, 50 or 100 layers of cells. Right, exactly. So you can lose like 100 layers of cells and you don't even notice. That's cool.
And it helps reduce the flow.
Right. You could also say something about one of its functions is temperature regulation. So it produces sweat to do that. It has sweat glands, which is how the structure supports it. Anything else from 5? What about 6?
The osteoporosis, is that the same as
Yeah, so osteoprogenitors, so it's chapter 6b there. They're bone stem cells.
Okay, is that the same as the?
So the osteoprogenitor, they're going to turn into all the other bone types, like osteoblasts, osteoclasts.
So they can go to be either one of them?
So they're just bone stem cells, basically. The last one?
Yeah.
Yeah, so let's contrast compact and spongy bone tissue in terms of structure and function. So we do structure first. So which one has osteons? Compact or spongy? It's compact has osteons. And which one has trabecula? Spongy there. And which one's stronger? Compact. So it's kind of a structure one. And then function. Compact is, for strength mostly and support. And then the spongy kind of protects the bone marrow, either red or yellow. Well, it could be red, I guess. All right, what about 7? List the 5 types of bones based on shape. Long, flat, short, irregular, and sesamoid.
What are the sesamoid ones?
Like your kneecap,
Okay, like the
patella.
Aren't they the floating ones? Which is
Yeah, I mean, they
hyoid?
do what?
Isn't it your hyoid too? Is it a ring? Or is that anywhere you would?
I don't know, actually. Long, flat, short, irregular, and sesamoid. My guess would be flat for the hyoid, but I don't know for sure.
Really?
I don't know. It's pretty. All right, what about chapter 8? Which one? Explain how the structure of the pubic symphysis supports its function. So it's made, does anybody know what it's made of? Pubic symphysis? So it's made of fibrocartilage, which is really good at resisting compression. And whenever you're walking, there's a whole lot of force being placed on your pelvis, and it absorbs that force.
So if there's like high line, it wouldn't like go back.
It would just be squished, and then your two pelvic sides would be banging together. That would probably hurt.
So can you like, you know how you can wear down like your tendons and stuff? Can you do that?
I guess so.
Wear down?
I've never really heard of it wearing down, but
Or like how you
it can break from like childbirth or like too much stressing or trauma. It could be like separate, which probably really hurts too.
Oh, I have one question. What's the difference between the, I know it says hip bone, but the costal and the sacrum, like what are the, I know the sacrum is like the bigger part of the hip bone.
Both of those are part of your spine.
The sacrum's the very, the triangle pointed one, it's like a tailbone.
Yeah, so the sacrum's at the very bottom.
Well, yeah, but if you're, is that the
Oh, the
bottom?
sacrum's like the little like,
one above it.
oh,
Mhm.
okay, I've got those mixed up.
All right, what about 9 part 1?
So describe the following movements.
Well, which one are you not sure about? And remember, I would say if this is on the test or something, like move yourself around. I bet you know almost all of them, just if you actually like thought about it and moved some body parts around.
Hyperextension is basically backwards, right? Because you're bending it rather than what you're actually.
Not necessarily. So let's do your shoulder, for example. What would flexion be of the shoulder? Right? So flexion, wait, do it forward, sorry. So that'd be like flexion, or is that backwards? Flexion, extension, hyperextension. Right. Not all of your joints do hyperextension, but some of them do.
It doesn't feel good.
So lateral flexion, that's like this one. Wait, I'm so bad at these. It's so embarrassing. This one. There we go. I need to study this chapter some more, clearly.
What's the little cartilage thing it's called? The little cup that your shoulder sits into?
The labrum.
Is that what that's called? It's like that little.
Mhm.
Yeah, it's like forms that socket. You don't have like a socket there.
Anything else from chapter 9? Or what about chapter 9, part 2?
Can you give me an example of the saddle?
Oh, what?
A saddle joint? It's on, well, because A is like the 6 things. Are those like the different types of joints?
No, the 6 factors that influence it. So like the shape of the bone, how tight the ligaments are. So that's chapter 9, part 2A there. So bone shape, how tight the ligaments are, how tight the muscles are, if there's any soft parts in the way, which means like basically other body parts in the way,
So that like you can't.
right? Yeah. Or like your shoulder, right? Your shoulder could go farther in, but the rest of your body's in the way. It just stops it. And then hormones and disuse. And while we're on that one, why don't joints work as well as you get older? One of the things that was kind of in the joint section, less synovial fluid, but also the ligaments get tighter as you get older. And they don't stretch as well. So what structural factors permit the shoulder to allow more movement than the hip? Right, but what about the structure makes it less stable? So is the hip. What? So the hip has a deeper socket. The shoulder has a shallow socket. And which one has tighter ligaments, do you think? Stronger and tighter ligaments? The hip has. Exactly. Well, certainly which one has bigger muscles around it? The hip, right? It's more stable, it's got tighter ligaments. Right. It's less movable. But the structure parts are the size of the socket and how tight the ligaments are.
I didn't read that question. I didn't see
Oh. What about chapter 10, part 1 there? Describe how muscle fibers shorten to produce a contraction. So, do y'all remember anything about that? Hopefully. So you've got, the sarcomere thing that you drew, the actin and the myosin overlapping like that. And then the myosin, and it's all attached together at the end, right? And then the myosin pulls on the actin fibers to pull everything closer together. The type, Yeah, that is true, but you don't need to know anything about Titan for this one. And describe the 4 special properties of muscle tissue that enables it to function correctly. We've got 3 E's. So elasticity, which means it's elastic. Extensibility, that means it can stretch without breaking. It's excitable, which means neurons can activate it. And it's contractile, it's able to shorten. Contracting or contractible, contractility, I can't remember what it says exactly, but contractibility, it's able to shorten when told to. Right, that's one of them. So we've got elastic, extensibility, contractility, and the excitability. So extensibility means it can stretch without breaking. Elasticity means it goes back to its normal shape after you stretch it. All right, what about part 2? Describe the role of calcium in excitation, contraction, coupling. So whenever the nerve activates the muscle, it eventually leads to calcium being released. Does anybody remember what calcium binds to? So calcium binds to troponin, and troponin is like in the way.
Is that the process wrapped around the troponin and then the calcium making it move so that it can bind to the receptor site?
Right. Yeah, so you've got troponin that's normally covering up actin, so myosin can't grab it. Calcium comes in, and it tells it to move out of the way. So troponin moves, and now myosin can grab actin.
Troponin's wrapped around that.
Right. So it's like covering it up, it's like blocking it, so myosin can't grab it. And calcium binds to troponin and tells it to like rotate or move out of the way. What about 11? Which one? Oh, I was like, I don't see an E on 11. All right, what about 12 then? So explain the events of signal transmission at chemical synapses. Maybe I'll pull up that little diagram, might make it easier. So that's chapter 12. Some part. All right, so the chemical synapse. Remember a synapse, connection between two neurons. So that means the neuron that the signal is coming from. Is that the axon terminal or the dendrite? It's the axon terminal. So the signal arrives, which you remember is positive ions, basically. And it gets there. It causes voltage-gated calcium channels to open. Calcium comes in, causes neurotransmitter to be released. Neurotransmitter comes over here to the dendrite, and it's going to open ligand-gated sodium channels on the second one. So you've got positive ions coming in, opening a voltage-gated channel, which causes neurotransmitter to be released. That neurotransmitter is like the actual molecule that goes from one neuron to the next one. And then it binds to another receptor there, ligand-gated. So neurotransmitter binds to that receptor and causes it to open and let positive ions into that next cell.
So once a certain amount of positive ions move down and come into the terminal, it opens calcium.
Right. So it opens the calcium channels at the end. You don't have these channels like in the axon. You only have them in the terminal there. Yeah, and I think on the slide before it's got it described too. But that's the picture one.
And then so basically how the action signal is propagated down in neuron axon is just once a certain amount of sodium reaches each part, it just causes the gates to open all the way down. And then, but the sequence of events of it starting.
Yeah, I'm confused on the
Sequence of events is.
Yeah, so chapter 12 for C would come. Oh, that's weird. That's why y'all are confused. So D comes first, and then C comes second.
See, yeah, I'm just really confused on, because on the test, I think we got both of those, and I was having trouble, like, differentiating which one was which.
Yeah, so for D, the sequence of events that makes an action potential, that's happening at the dendrite. And so, if it's, depending on what the first receptor is, could be ligand gated, could be voltage, could be mechanical, but something opens up some sodium channels in the dendrite. So let's say it's the mechanical one. If you're pressing on that neuron, it forces the channels open, and it lets sodium ions in to the cell. And if it gets enough sodium ions in there, that generates an action potential. Which remember is that threshold at minus 55 business. So D happens at the dendrite, and then once you make the action potential, then it goes to C there, which is how it moves down the axon, which was simply the positive ions opening up a voltage-gated channel, which lets in more sodium ions. And then that opens up the voltage channels next to the sodium ions. It just goes step by step. Same thing over and over again. Does that make more sense?
The one, that last part, that, so then,
Yeah, you could think of it as like little dominoes getting
opening
knocked
the
over.
ions and letting more.
Right. But at a different spot. So like, if you're traveling down the axon like this way, first the sodium ions come in here, and that makes this gate open next to it. And this sodium ion here makes this gate open. So now it could come in, which opens up the next hole, kind of like that. I know, I don't mean to put them backwards. Just, yeah, you definitely want to write down on there that D comes before C. And just like I told you before, if you feel like you're having trouble with chapter 12, it's probably because you're confused about ion channels in general. So go back and review like just how they open, what they are, like the beginning of that PowerPoint. Because that's usually where people are getting stuck. What about 13? Yes, so stretch reflex. It's got two functions. The first one is to prevent muscles from stretching too much and breaking. And then it's also for your posture. So like when you're leaning over this way, it tells the muscle that's stretching to contract so you don't just fall over. So it kind of helps with your coordination and balance and everything. Because any stretched muscle contracts and it'll keep you sort of stable. The tendon reflex is kind of the opposite. So it prevents muscles from exerting too much force and damaging the muscle or the tendon. So, you're doing some bicep curl with like 300 pounds, and the tendon that's connecting your bicep to the muscle senses that's too much force, and it makes the bicep relax. Right. Prevents too much of a load. Because if you tried to curl 300 pounds, you'd probably like tear your muscle, tear the tendon, who knows. But they both stretch and tendon to protect the muscle just from different things, right?
The next one. Lumbar and spinal cord.
Yeah, so explain why the lumbar spinal cord is more gray matter than the thoracic. First of all, for gray matter, what's the difference between gray and white matter? White is axons, gray is like dendrites and cell bodies. Synapses, so down at the bottom of your spine, there's more synapses because it has to connect to all the neurons that are going to your legs and everything. And there's not that many axons because they all branched off already higher up. But once you're up here, you've got all the axons carrying information from everything below. And the only gray matter there would be the things connected to your arms or something. The reflex arc. Yeah, let me just pull up that picture. That's 13. Towards the end. All right, so 5 main parts of the reflex arc. You've got the receptor itself, so that's like the dendrite, basically, of the first neuron. And then the neuron that takes that information, it's a sensory neuron. Integrating center decides what to do about it. The motor neuron takes the signal from the integrating center, and then the effector actually does the response. It's like contract a muscle or relax a muscle usually. Yeah. That's the part that decides what to do about it. Yes. So it's right here in the spinal cord in the central nervous system. Other reflexes might be in the brain or something, but definitely in the central nervous system. The integrating center decides what to do about it. Because maybe the stimulus coming from here isn't great enough to have the reflex happen. All right, what about 14? So C, the structure and importance of the blood-brain barrier. So we've got 3 layers, the blood-brain barrier. You've got the tight capillary walls with tight junctions. Then you've got the basement membrane and astrocyte feet. So you got these 3 layers to keep, dangerous stuff in the blood and out of the brain, away from the neurons. That's the importance of it. You see, catch that? To just keep most everything, keep anything dangerous or toxic in the blood and not touching your neurons. basically. So those specialized capillaries that are very tightly packed and everything, anywhere you have blood vessels in your brain, you have the blood-brain barrier. So it's not between your skull and your brain. It's between the blood vessels and the nervous tissue, which is everywhere. All right, what about 15? Part 1. Oh. Yes, neurotransmitter. So describe the neurotransmitters and receptors involved in sympathetic nervous system response. All right. How many neurons are there in a row for the sympathetic and parasympathetic? 1 neuron, 2 neurons, 3 neurons? Remember that diagram? I hear some people whispering too. You're right. What's the first one called? Post. And then you've got like the effector, right? So preganglionic, it sends out a neurotransmitter, which is acetylcholine. And then the postganglionic has a receptor. What receptor is it? Nicotinic receptor. So the preganglionic sends out acetylcholine. Postganglionic has a nicotinic receptor. And then the postganglionic, it sends out a neurotransmitter. That's not going to be any room. Norepinephrine, or NE. And then what's the receptor for that? So the effector has the adrenergic receptor. And that was nice. I only put the sympathetic one on there for neurotransmitters and receptors. So you've got preganglionic neuron, releases acetylcholine from its axon terminal. The postganglionic dendrite has a nicotinic receptor. And then the postganglionic axon terminal releases norepinephrine, and the effector has an adrenergic receptor. Yeah, so a signal would be traveling through all of those steps, basically. Do I? It's a cholinergic. But that's not on the list, so you guys don't have to worry about it. But yeah. Do what? That's in the parasympathetic. Oh, I think that was that the sweat gland exception? So just worry about this one. This is almost all of them.
Basically what that's saying is that your preganglionic neuron releases acetylcholine to the postganglionic neuron, which has this nicotinic receptor, and that releases norepinephrine, and that's what causes the effector to do whatever the effector has to do. How do you have to say that?
Yes.
Okay.
Got it. Yes. Yeah, my sinuses are so messed up. I can't. I just stopped being able to hear out of this here.
I feel so echo back to that time.
This is weird. Talking to my good ear. Anyways, what about part 2? Where they explain the relationship of the hypothalamus to the autonomic nervous system. So the hypothalamus decides when to activate the sympathetic or parasympathetic nervous system. All right. Study well. I have some office hours tomorrow morning.